So this video is just going to be a quick overview of the variations among single genes and multiple genes. So variations among single genes, one type of variation you can find is incomplete dominance, where two alleles may produce an intermediate phenotype when both are present, rather than one fully determining the phenotype. This usually corresponds with like blending of alleles, right? And this can be seen in snapdragons, where a red snapdragon crossing with a white snapdragon usually makes an intermediate color, which is pink, right? So that's where R and white, the red color and white color are both found, and they blend their phenotypes. Codominance is where two alleles simultaneously express their phenotype without one fully determining the phenotype. And this can be seen in blood types, where there's A blood type, B blood type, O blood type. A and B are both dominant, but O is recessive. So B is dominant to O, B is dominant to O, A is dominant to O, right? Since O is recessive, but A is not dominant over B, B is not dominant over A. They're both equally dominant, and when they're found in the offspring, they're usually seen together. And they make a whole different blood type, which is called AB. So right here in this cross, we're crossing an individual with AB blood type with an individual with A blood type, and their genotype is AO. So when we do that, we get one individual with A blood type with both alleles being for A blood type. Then we have individual with A blood type, right, with one allele being A, one allele being O. And in this individual right here, we have both A and B. They're both dominant, so they're both seen in the blood type, and thus they're called AB blood type. And this one, B allele with O, that's B blood type, and B is dominant, and this one, A is dominant. And codominant blood types can also be an example of multiple alleles, where there are 3 or more alleles involved, right? So ABO blood type is another example of that, where multiple alleles are involved, and there's codominance involved, and there's basically multiple alleles and codominance can be looked at interchangeably. And the example too, it's the same example, ABO blood types. Now, lethal alleles. Some genes have alleles that present survival when homozygous or heterozygous. And in this example, homozygous recessive basically just means that the offspring will just die, right? That it has an allele that causes death. So in this mouse right here, when this was crossed, when these two parents were crossed, it produced one offspring with 2 homozygous recessive alleles, causing that offspring to actually die. And all of these things, multiple allele, codominance, incomplete dominance, lethal alleles, they're all involving variation, right? And mutations that happen during meiosis, happen during independent assortment, and stuff like that. Now, epistasis, this is when the alleles of one gene mask or conceal the alleles of another gene. So they often arise between 2 or more alleles, and 2 or more different proteins participate in a common cellular function. So in this example, a colorless precursor via enzyme C creates this intermediate, and this intermediate can be changed into this purple pigment via this enzyme P. So there's obviously 2 different genes right here, and this gene right here just masked this intermediate, right? Masked this intermediate. This gene made this color right here, right? But since we have the interaction of another gene, it made this colorless intermediate purple. And this example can be seen in flower color right here. So homozygosity for the recessive allele of either gene results in white phenotype, thereby masking the purple. So right here, this is the example right here. And both gene products encoded by the wild type alleles C and P are needed for the purple phenotype. That means that this gene and this gene are both needed in order to make this purple pigment, since the first step is basically this gene coding for this colorless intermediate, and then the next step is this gene masking the effect of this colorless intermediate and making a purple pigment. Now, plyotropy is when the gene product can affect cell function in more than one way. So basically means that the gene can be expressed in many different cell types. It can be expressed in different stages of development. So it means that one mutation, one gene can affect many different parts of the body. And this can be seen right here in phenylketonuria, where this gene right here, this process right here, affects many different parts of the body and systems of the body. So that's plyotropy. Now, types of mutations. There's some more extra type of mutations where the first one is the loss of function mutations, where a new phenotype results from a change in activity. And mutation causes loss of wild type function. Gain of function mutation means that the mutation enhances function of the wild type. Neutral mutation, there's no change in the phenotype. Examples of these are, for gain of function, a protein encoded by the mutant gene is changed so it gains a new or abnormal function. So it's gaining more gene product. Thus, it's adding on function, right? It's giving that certain protein or gene enhanced function. We also have for dominant negative. In this example, this is an example for loss of function. Actually, no, not a loss of function. This is actually a gain of function as well, since the protein encoded by the mutant gene acts antagonistically to the normal protein. It's still enhancing the function of that certain gene, just in the opposite direction. Now, Haploin Insufficiency is a example of loss of function, and in this, the heterozygote does not make enough product to give the wild type phenotype. So that means that the gene product is lessened, and thus there's a loss of function. So these are all variations that can happen in meiosis and independent assortment, and you just need to know them, since they have the process of meiosis involved in them. And yeah.